Thank you for the introduction and good afternoon, everyone. I'm very happy to be here in person and also would like to share with you about what PBS Biotech is going to do, as well as how we're going to really help the cell therapy industry, especially for their commercialization. So let me see. So the company's mission is we're trying to improve patient lives of a patient by helping them to be successful for commercialization. And as you notice that there's tremendous progress is made in a scientifically demonstrate the efficacy and all those potential the therapeutic product. But now the challenge is, especially for the allogeneic application, the capacity of manufacturing can be potentially a bottleneck. And then how can really enable this and then allow that all this innovative product to go all the way to every patient. So our vision is we try to provide the best the manufacturing platform as an industrial standard, as well as unsurpassed the technical the services, and then to unlock the clinical and a commercial stage of allogeneic manufacturing and the cell therapy manufacturing. So recently, it's especially during last several years, it's made a tremendous progress. I know everybody heard about the gene therapy as well as autologous the immunotherapies. However, they're also very interesting, the progression in the allogeneic cell therapy. For example, about a year ago, the Vertex announced that the first in-human clinical trial outcome for the type 1 diabetes using pluripotent stem cell-derived the beta cells, islet cells. And then in the beginning of this year, the CRISPR and Avaya side, they started a new clinical study for first in human using gene-edited, which basically the suppressant, the treatment is not necessary because it's a knockdown gene and the pluripotent stem cell. They generate the beta cells from those pluripotent stem cells. So all this kind of approach is really targeting for mass, the number of the patients in the US alone for the type 1, type 2 diabetes is more than 10 million patients. And how are we going to support those kind of a mass production of those pluripotent stem cell derived the beta cells. Another example is also the very growing the attention as well as the progress in immuno-oncology is starting from the pluripotent stem cell. Historically, this immuno-oncology is starting from the donor cell derived, but as you know that the T cell and then NK cell is not necessarily prolific. They are the final terminal, the functional cells, and whereas the pluripotent stem cell can grow very fast and in a large amount. So their approach is, for example, like the Century Therapeutics or Editas, they're using pluripotent stem cell, expanding in a large volume, and then differentiating into their specific target cells. And during that process, they also integrate this gene editing process to minimize the immunogenicity challenges as well. So the immuno-oncology is another growing the area. And again, same challenges. This is an allogeneic approach, and of course, including CAR-T, and the subsequent modification can improve the efficacy. But the bottom line is how we're going to mass produce this type of immune cells from pluripotent stem cell can be another challenge. Another area we just heard about is the bioengineered human the organs, but in this particular case is actually not from this organ cell recirculating, but is starting from the patient own pluripotent stem cell. And then differentiated into, in this case, is a cardiomyocyte, use the pig the scaffold, the heart scaffold as a basic structure, and then covered and printed with his own patient PSC-derived cardiomyocyte cells, and then creating bioengineered autologous heart. So this kind of approach is all required a large amount of pluripotent stem cell expansion as well as differentiation. So going back to a little bit of biology, in the cell type, there were most of the allogeneic application as a typically two major type of cell types. One of them is a pluripotent stem cell, whether it's iPSC or is embryonic stem cells. This type of cells grow and differentiate as a cell aggregate. You can see in this picture, even though we inoculate it in single cell suspension in bioreactor, and as they grow, they actually aggregate and then create this sphere shape of a form. And that is very important for their differentiation because they communicate with the neighboring cells. And then currently we estimate about 120 companies are developing this type of a drug modality and the PSC-derived therapeutic product. And then PBS is working around 78 of them. And the second type is of human primary cells, like a mesenchymal stem cell or a stroma cell, or chondrocyte, and then some cases the exosomes, which this type of a primary cell is because it's anchorage dependent. They grow on the surface of microcarriers, which is a plastic beads, can be circulated in suspension in the bioreactor. And then you can see in the picture that the blue color is the plastic beads, and with the green fluorescence is the stained cells attached on the surface. So they can actually grow and during the suspended in the bioreactor. And we also estimate about 120 companies are developing human primary cell-based therapeutic products, and then PBS is working with around 9 of them. So all this type of cells, in order to mass produce, that the 2D culture platform is commonly used in the R&D and early stage research, but it's not scalable, and also it's not economical. So a lot of people then obviously thinking that 3D culture is a solution because we have a very good experience from the biotech industry. Monoclonal antibody use the third type of bioreactors, and then they scale up to 2,000 liters, and the stainless steel bioreactors even 20,000 liters. So that's an obvious choice that we can use a 3D bioreactor system. However, one challenge of that approach is going to be that the cell conditions for the biotech industry for single cell suspension, like a CHO cell or E. coli, is completely different than the human cells. Number one, the human cell itself is a product for cell therapy product. Number two, the human cells are isolated from the organ, and so they are anchorage dependent. They have to attach on a certain solid or neighboring cells. That's the reason why they're growing as either aggregates or microcarriers. And then because of this, the particle size is much bigger, usually around 200 to 400 micron diameter, whereas the single cells, the diameter is average about 15 to 20 micron. So you can think of one-dimensional 10-fold. The 3D mass is 1,000-fold bigger mass, need to be suspended in the bioreactor. The way to suspend it, you need to increase the agitation speed, which will create more shear. And then the human cells can be shear sensitive, respond to this shear environment in the bioreactor. So the hydrodynamic condition has become a challenge for the human cell growth in suspension in the bioreactors. And then another challenge is the small scales are relatively easy to the suspended dose, but when this volume, the size of a bioreactor has become the larger, and then that's where conventional bioreactor become the more difficult challenges. So that's where this PBS vertical wheel bioreactor coming as a solution to overcome this kind of hydrodynamic challenges. So we have a very unique vertical wheel bioreactor, and you can see there's a very uniform homogeneous distribution of the right-hand side is actually microcarriers, the suspension during the slow motion in the bioreactor. And we provided this family of different sizes of bioreactors, which is 3 liter, 15 liter, 80 liter is a standalone, a fully controlled. The bioreactor was a mini 0.1, 0.5 is designed to be used in an incubator in the R&D lab. We also provide process development contract services, not only the equipment, so we're trying to optimize the process for the client as well. One key point is that the uniformity of aggregate size for pluripotent stem cell is very important. So when the cells grow in a high energy dissipation rate environment, a high shear environment, the average size is getting smaller, and if they grow in the low agitation condition, it becomes larger. The reason why that the uniform size is important because during the differentiation process, you need to change the growth factors in media and diffusion rate among the different size of aggregate. It's difficult to synchronize the timing of a medium exchange. So if you grow in the vertical well bioreactor, very homogeneous EDR condition is end with a very sharp peak, and then whereas a conventional stirred-up bioreactor is a flat or bimodal distribution of a cell aggregate. That's the challenges. So what we did the CFD analysis, various sizes of bioreactors. We identified a certain EDR ranges is optimal for the cell aggregate formation. If it's below, that is two big clumps. And if it's too high, then they are not uniform aggregate size. So we identify those hydrodynamic conditions of the various bioreactor sizes. We apply that, and for example, this is a scale-up data from 0.1 liter, 3 liter, 15 liter, using perfusion process, pluripotent stem cell to the 4 million cells per mil. And there are 3 different sizes of bioreactor. The cell growth profile is almost identical. And then we also scale up this 80 liter scale. As you can see that the left, the column is showing the 2 liter the roll-up bottle process, current process from our customer, the ViaCyte, and the courtesy of their data, and a different process of differentiation, and then we scale up to 30 liter and 80 liter and maintain the very comparable aggregate sizes. So that demonstrates that even at the 80 liter scale, the size can be maintained for the 2 small liter bioreactors. So we have selected customers that I mentioned that are almost 2/3 of industry customers as well as academic collaborators. Another final point, and I want to mention that we also collaborate the partnership with the Stem Cell Technology, which is the cell therapy media company. And they launch our mini product for the R&D, especially academia labs. And then they're encouraging to scale up the suspension mode in our mini bioreactors. And then we will also continue in collaboration to generate data for the larger scale bioreactor operation. So I'd like to pause at this point. I'm going to play the company introduction, the overview video briefly. So.
PBS Biotech makes a full range from 0.1 liters up to 80 liter bioreactors, really for mammalian cell culture. More importantly, PBS Biotech provides the services to help enable the success of our customers for scaling up their process. The unique feature of the PBS single-use bioreactor line is certainly the vertical wheel impeller. What that impeller provides for you is a very homogeneous, very low shear environment, and the same environment at the 0.1 liter as the full production scale system.
There's 3 main subsystems that put together make up the PBS bioreactor. First, we have the disposable vessel where the cell culture is grown in. The second is the control system, the software that actually drives everything, controls all aspects of the cell growth process. And then you have the electromechanical system of the bioreactor itself, which includes the sensors, the pumps, the motors. All that put together comprises the bioreactor system.
We have very experienced and dedicated people that understand GMP regulation and compliance requirements. We have bioreactor assembly in-house. We have single-use manufacturing in-house, QC, warehousing, shipping, distribution, customer service. So we are a full integrated unit that can communicate, work, coordinate, and serve better our customers.
Customers don't want just a product manufactured. They really want a service. They really want the entire solution. That's what PBS strives to be, as a solutions provider. PBS is innovative. We're a fast-growing company. We develop high-quality products that work well with a variety of stem cell applications. And we have a great service team that will continue to just support you every step of the way.
Innovation should not stay as an idea, but innovation needs to deliver all the benefits to the endpoint user, which is a patient. I think the best benefit the PBS can offer to our customers, especially in cell therapy industry, is to allow to scale up their R&D performance data in clinical stage manufacturing as well as a commercial stage manufacturing, so they can actually close the gap between the R&D to the commercial. And that is a potentially big bottleneck for the success of the cell therapy industry. And that's what we're trying to provide a solution to this emerging, exciting opportunity with this innovative therapy product.
PBS Biotech will enable our customers to treat the thousands and millions of patients who can benefit from cell therapies to cure cancer, to cure diabetes, to cure chronic heart failure, and other life-threatening conditions around the world.
Thank you for your attention. I'd like to, I know what time is already passed, so we probably cannot take a question, but I will come to discussing outside. Thank you very much.